Hey there, Caleb. Um, so I'm going to try to make this short. Okay, so there's two little ideas. And so the first one combines rolling cycle replication. I'm sure you know what it is. I'll just give you a quick overview. So you have a double-stranded plasmid, and then you have a nickase, and it makes a nick in one of the strands. So it kind of pops it open, and then it starts replicating, like here. And because it's replicating, I think it uses one of these strands as a primer actually for replication. As it replicates in one direction, it actually kicks one of the strands out. And so what you end up getting looks something like this, where you have single-stranded versions that are kind of connected, and normally in a virus, and these kind of would be packaged into different viral particles, but we want to use them for a different purpose. So I'm interested in just this little region here. Because I want it to do strand invasion. So strand invasion in a homologous chromosome, or a homologous sequence of a chromosome, series of chromosomes. So we have a region of DNA here, and so this is something called a D loop, and it's induced in strand invasion in a REC-A background. REC-A is kind of the protein that takes single-stranded DNA and it guides it to its double-stranded homolog. I don't know how it does that, but it does all the time. I guess I should have colored this a different color. So let's say this one's red. So basically this, thanks to REC-A, will just kind of find its homolog and it'll bind as if it was about to do homologous recombination. But because it's single-stranded, then it's just, this is all that's going to happen. And so of course what we'd like to do is then, we'd be able to map, like, oh, where's this going to reach up to, and then exactly where it reaches we make a cut. Exactly where it reaches, we make a cut, and so then this guy gets sent out, and it gets chopped up by exonucleases, and then we take our ligase, of course, and we ligate from here onto there, and from there to there, and this guy onto here, and then essentially you've just reformed. So you have like that, like that. So this is your new DNA. And so what I think is good about this is that, this here is supposed to be your young DNA, and it has fewer errors than the old DNA. And so then one worry would be, okay, so great, you've got your young DNA here, and it's got its little differences from its old DNA. Let's say the old DNA has a mutation here, a mutation here, a mutation here, whatever. But the young DNA is great. The problem, though, is that the old DNA also has methylation, right? So it's methylated in a certain way, and when the replication machinery comes, or sorry, the corrective machinery comes in and it looks at this old strand and says, like, this is not, these base pairs don't match here. These base pairs don't match here. And then it's going to say, which strand do I believe? It's going to believe the old strand. So what we need to do is find a way to either methylate the young strand as well, or demethylate the old strand. It's a problem which I haven't really gotten to. But anyway, so then taking that to kind of a larger scale, if you look at something like bacteria, so the idea basically is to do it twice. So if you use an origin of replication, two of them actually, which are separated by something like 50,000 base pairs, 50 kb, and then you do this kind of single-stranded invasion at both of these sites, except that these strands would actually be 50 kb each. Or maybe not. Maybe it could just be, I haven't really worked all this out, maybe it could just be this part here, and that would only have to be 25 kb, and then it's only homologous to this region in the middle here. And so the goal is to kick out the entire region between these two ORICs, the chromosomal origin of replication. Right, and so we kick this guy out by the same method that we did this here, except that it's kind of on a larger scale. And I was reading, and it actually turns out that the holiday junction that is produced when you do a strand invasion, it naturally, if this is larger here, it'll kind of naturally push itself kind of over in both directions actually. So, and by that I mean, like once the DNA is broken open, then then if there's more strand here, it kind of pushes it open in either direction. It's a loose explanation, but anyway. Yeah, and so then you can imagine how if you did this with many single-stranded homologs in a kind of an older cell, using young single-stranded homologs to fix not broken, but you know, genes that code for proteins that aren't working anymore in reactive oxygen species. In dealing with reactive oxygen species, then maybe you could do something about all of this. Anyway, so that's the basic idea. Yeah, so I guess eventually you'd use like a big virus, something with 2 megabase pairs or something, and you know that the viral insert would be divided up into, let's say, I don't know, 1,000 of the most important genes. And then of course this would do rolling cycle replication, kind of like before, and then each of these segments would be divided into, you know, each of those little genes. So I don't know, maybe there's some kind of restriction enzyme that we can include that would cut all the little sites in between, and then each of those goes and seeks out its, you know, its homolog in the chromosomes, and it fixes it. So yeah, so that's the idea. I look forward to hearing your feedback.